So yeah, if anyone has any questions, please come up to the mics and fire it. Oh, I see people moving already. Brilliant.
Questions, yeah.
Okay. Yeah, do you want to start us off?
So thank you very much. I'm Gal Keshet from the Hebrew University. I have a question about the DNA data storage. So in order to access the storage, you need, you said like that you need to perform like the sequencing and then you can read the data. But every time like you need to sequence the whole thing, how do you access specific places of the data?
This is a really good question, thank you. So some researchers are working on what they call random access. So how can you access to actually some specific information? So for example, if you have a tube of a lot of different files, usually the sequence of DNA will include an address, so a barcode explaining what is this information about. And you could actually do a kind of random access. For example, if you try to amplify this DNA, you could use a specific primer for this specific information and amplify this specific file among this tube.
Thanks.
You're welcome.
Hi, Aaron from Australian National University. I also have a question about the DNA sequencing stuff. So the speed that you quoted at the end there for storage was 1 bit per second. And if I think about that, even roughly in terms of terabytes, it seems like that's still many, many orders of magnitude off being somewhere useful. I was just wondering, what are the prospects for actually improving that speed? And is it just a cost thing, an engineering thing? Like, do we know how to do it? Or is it something fundamental that still has to be sort of done?
Thank you. To start with, like I know you tried to compare that with I don't know, hard disk writing speed, but actually the thing is that DNA is more aiming to store some specific information, so not any kind of information. It's more an archive thing. So actually among data, they used to differentiate what is hot data and what is cold data. So hot data is the information you always want to access to and that you want to edit. And cold data is usually some information you're not really interested in, or sometimes they're really important, but you want to keep them for a long time. And actually, like 75 of all the information we store is cold data. So actually, it will be very useful to like store all of this information, 75% of all the information we store into DNA. So even if it is slow, it can still be used for that.
Thanks.
Hi guys, thank you for your pitches. I also have a question for Paul. My name is Barbara, I'm from Czech Republic. Yeah, that's what I forgot to say. My question is, and I'm sorry if I didn't catch it during your talk, but what precisely is your part in this whole topic?
Yeah, thank you. So it was a really broad presentation to explain you the what are the important steps for DNA data storage. So I'm an engineer, and I actually still work on the part that is about how you do the synthesis. So there are many ways that are explored in my lab, but specifically what I'm trying to do for synthesis, like to start with, what is phosphoramidite chemistry? It is called de novo synthesis. It means that you're adding one base after another, basically. What I'm trying to do is to do like a block synthesis. So if you would need, for example, just an analogy, if you would want to write a text, like the de novo synthesis is like writing it letter by letter. My idea is more writing it words by words. So like you need a kind of dictionary to access these words. So this is called a library. So what I'm trying to do is to make this kind of library with a lot of different words on the surface, and put that in a microfluidic system, and then try to catch these specific DNA strands, and then write the information you want.
Thank you very much.
Yeah, thank you.
Hello, my name is Bahar. I have a question about the DNA sequencing as well. So I When you were talking, I was imagining. So, for example, I have this photo on my phone. I want to put it in my cloud, and then if I'm 2 continents away, I can just like log in another laptop with my account in the same cloud and see that image. But if that image is stored in like as a DNA, then it's stored somewhere. Then I wanted to access that. Like somebody's gonna go and kind of decode it and then give me the result. And like it takes a lot of time for me to just access one picture. So I don't think it would be used for populations. Am I right?
Yeah, it's a bit like my answer would be very linked to the previous question. Like it's not supposed to replace the hard disk or the cloud that we're using. It's just to remove a lot of information that is in the cloud, but that is useless to be in the cloud, sort of.
So basically for information that nobody needs anymore.
Yeah, for
okay.
information that nobody needs, but that is actually really important for financial records. You need to keep this information.
Yeah,
For
I
media
was just-
archives, you need to always keep some information of what is happening now.
Okay,
So
thank
it's still
you.
important, yeah.
I have a question to Dr. Kampun. Sorry, I'm Nurul, a PhD student in chemical engineering. So I'm interested in wettability properties. Then you mentioned that one of the benefits of your material is tunable hydrophilicity. So my question is, can you explain more how can you adjust the hydrophilicity of your material, and to what extent can hydrophilicity of your material be decreased and increased?
Yeah, no, that's a great question. So it really depends on your application and in terms of what kind of hydrophilicity you want, particularly with these aliphatic polyesters and polycarbonates, where they can hydrolyze and they can degrade. And that is a benefit in some applications, like in vivo. But if you talk to anyone in industry, biodegradation is like a no-no. They do not want materials to degrade and properties to deteriorate. So in this case, we changed the content of polyethylene glycol, which is sort of the gold standard biopolymer. And the more content we put in, the greater the water uptake. The swelling index only went up to like a few percent, so it was still marginal because the majority of the backbone is very hydrophobic. But that small increase is enough to have a big effect on the rate of hydrolytic degradation. There are other ways. There are so many handles in these kinds of materials we can pull to change different properties. So we could change the crosslinker, we could change the backbone. There's an N-butyl side chain on each repeat unit of this polyester block. If we remove that, we could change it. So there's all these different handles and levers we can pull depending on the application. And that's the key. It's really application dependent. I hope that answers your question.
Thank you.
Cool. Thank you.
Hi, nice to thank you for a great talk. I have a question for Dr. Kam. Around the last slide, you show us the fluorescence image of the cell, and then the cell with the degraded part sample looked shrinking compared to others. So, but you said there was no cytotoxicity. So, did you check some cell viability or some another cell mass assays?
Yeah, so that's a good point. We're unsure on why the cells, we think it's a surface interaction effect that was resulting in the cells being different sizes. And like appearing as different sizes, I should say. I'm not a biologist, I'm a synthetic polymer chemist, so this was not my specialty at all. But we did a live dead assay and we didn't see any cell death. That's just the beginning of the story. It's opening up. It's indication to us that we could pursue this further. But at the moment, yeah, we're not seeing any cell death, so we could use it in that application. That's what we're going to look at going forward. But no, you raise an excellent point, and it's something that we want to study further now.
Okay, got it.
Thank you.
I thought when the polymer degradation happened, the condition of the medium around the cell maybe change, like pH change, and then maybe that affect to the cell condition. So I thought maybe that affects something.
So I should have been clear, I think. The degradation was separate to the cytotoxicity. That was just done on glass slides that we could spin coat the polymer onto, and that wasn't degraded. The degradation is done in quite extreme conditions, partly for time constraints and because the material is, they degraded on a reasonable timescale, but we had to accelerate it a bit.
Thank you.
Should we go? Yeah, we'll go. I don't know who was first, but.
Hi, I'm Masaki from Japan. Thank you all for your talk. And I have a question to Hannah. So if I'm not mistaken, your gel or I don't know, your approach can be used to any type of cells. So I want to ask, why did you choose leukemia cells to apply your gel? And also, what's your approach to make your gel suitable to research leukemia cells? Is there any like specific way that you kind of like, I don't know, accommodate like, I don't know, like, is there any specific approach did you take to make it very, I don't know, make leukemia cells happy living in your hotel maybe?
So thank you for a question. So the first thing, why we chose leukemic cells. The reason is that our department is specifically focused on research in cancer and hematology. So basically, the idea was that we will use this opportunity of interdisciplinary science, since we are like a research consortium of multiple institutes. We are in life sciences. We have also departments that are dealing with research in material sciences, and we kind of combine our forces. They created the materials, and we use the cells that would be an interest of our research, which was leukemia, because we knew that the research that we are doing is really complicated when we do it just in the flasks, and we needed some improvement, and this looked promising. What can be done then, and what was already done by another of my colleagues, was that you can take the materials and put them also, for example, into the mice. And there they are biocompatible, and since they are kind of like small sponges and they are kind of soft, they are also vascularized, like veins go in them and so on. So they can be like a nice first step that you accommodate the materials with the cells in vitro, and then you have the material and you put it into the mouse. So this is nice, and also what it can be good for is that, but we didn't do it yet, you can kind of exclude some candidates from clinical testing when you do the treatment first on the cells that are kind of in 3D, because in 3D they can have some different resistance to treatments than when they are just floating in the flask. But this is something which still has to be done, and this is some like work of future, and it hasn't been done yet, but this is really something that it can be used for.
Thank you.
Hi, I'm Josephine from the UK, and you did just answer the question I was about to ask about the translation into in vivo models. So I've got a different question for Hannah now, and it's about how did you manage to get heterogeneity and spatial distribution of the different tumor microenvironment cells within your scaffold? And were you able to maintain the morphologies of the cells, particularly the adherent cells within that scaffold?
Thank you. So the seeding of the scaffold was like the chapter number 1, which kind of took a lot of time because it's kind of hard to homogeneously spread the cells all over the material. So what we used like some different methods, but what we used to put the cells into the scaffolds, when the scaffolds were in the hydrogels, which are kind of like already waterish, we put the scaffolds in the tubes and together with the cell suspensions, and we rotated it on the horizontal axis, kind of like a London eye for the cells, and they tend to go into the pores. But it has some disadvantage because you need to kind of overload the scaffolds, because they tend to, as they tend to go into the pores, they also tend to fall out of it. So that requires a lot of material in the beginning. What we can also do is that if you have the lyophilized, the freeze-dried scaffolds, is that you just put the cells into the like really dry scaffold, and as you put the wet substance into it, it kind of spreads all over the place, and the cells are basically everywhere. What we don't know, for example, is how deep they are in the scaffold. Like we know for sure that they are somewhere where the laser can reach with the confocal microscopy, but we didn't cut the scaffold yet to go into the deeper levels. And now I forgot what was your second question.
It was more just to do with the morphology of the cells. If you were using adherent, like fibroblasts within your gels, were they maintaining their physiological morphology?
Okay, so thank you for repeating. Yeah, so with the morphology, the adherent cells, yeah, they retain their physiological shape, physiological shape, but if they didn't, we applied that adhesion peptide because sometimes they were just like a kind of slippery. And with the suspension cell lines, they are mostly like just kind of floating or kind of sticking to the adherent cells, so they are okay-ish. And yeah, this is what we saw, and it was okay. But it was kind of sometimes hard to make them stay in the pores. So this is something that I think every material scientist combining with the life sciences has to deal with. That they're basically like going away from the scaffold. Yeah.
Cool. Thank you very much.
Cool. Okay, I'm going to use my privilege as moderator to ask the final question to Matteo. You talked about the concern if neutrinos are too low in mass or have no mass. Is that purely a detector limit, and is there ways to improve on that?
You're asking if I'm concerned that no one...
Well, yeah, so so I guess like if there are advances in detection for the neutrinos, like how would that, like is that an area that's being worked on, and how would that affect what you're looking at?
Oh yeah, there is an advance in the detector technology, but the problem at the moment is that the state of the art of the detector can no longer be advanced. So we need to find new ways. That's why we have come in, but we are not the only one. We are trying to came up with new ideas to detect in a very precise way the energy spectrum of the electron in some way. So we are just one of the many possibilities. The first one that works is fine.
Cool. Well, thank you. So yeah, with that, we'll wrap up this session. Thank you so much for your attention. If you have any questions for us, please come and find us.